Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to today's webinar titled Functional Recovery of the Musculoskeletal System Following Injury, Leveraging the Large Animal Model, featuring Dr. Sarah Greising, Associate Professor of Kinesiology at the University of Minnesota. Today, she will discuss the current pathophysiologic understanding of the skeletal muscle remaining following traumatic musculoskeletal injuries. I want to take this moment to thank our partners at the American Physiological Society and the sponsor for this event, Aurora Scientific, for making it all possible. I'm going to welcome Sarah to the floor. Hi.
Hi, thank you, Sydney.
You're welcome. Take it away whenever you're ready.
Got it. Yeah, thank you so much for the introduction, Sydney. And I too want to first thank you, but also the folks at Aurora Scientific and the American Physiologic Society for co-sponsoring this webinar. And with that, I will get started. So my lab is primarily interested in skeletal muscle function and physiology. And we look at that in the context of conditions that have a limited regenerative potential of that skeletal muscle. So this could be aging, this could be injury, disease conditions. But for today's talk, I'm really going to talk about volumetric muscle loss injury, or VML, as you'll see it abbreviated throughout this presentation. And with that, I'm going to talk about some of our work from small to large animal models, as well as how we've been pulling in functional recovery in this process also. So volumetric muscle loss injuries, although not new, the sort of clinical definition of it comes from this paper back in 2011. And this is the traumatic or surgical loss of skeletal muscle with a resultant functional impairment. And at the time, a little bit over 10 years ago, if we look at this bottom chart here, they have these possible therapeutic options. So these things on the outlook of the horizon of what people are thinking about. And one of which was regenerative medicine approaches. So as I talk through some of our data here today, I think we're kind of on the cusp of some of these, starting to get more beneficial for this injury population. We'll talk through that a little bit. So this injury, as I alluded to, is when a critical portion of muscle or potentially an entire muscle unit is removed due to trauma or surgery. We're seeing examples here from the literature of some individuals that this had happened from, but we know the outcomes, right? Persistent functional deficits, loss of range of motion, and disability that are going to be lifelong. When I think the key and something that we really focus on is this irrecoverable loss of the contractile tissue, as well as the strength of the muscle. And to date, at least, there's not a standard clinical or surgical or rehabilitative standard of care that's addressing that soft tissue loss or what is necessary for the muscle to regenerate. So if we look a little deeper into that one patient that's on there, we can use muscle function clinically and see what the function of this individual is. So keep in mind, this is after a number of procedures, surgical procedures to support bone union, which we can kind of see in those top radiographs. And after significant rehabilitation, what we're looking at here is isokinetic work loops as acquired from the biodex, so muscle function. And for this individual, his uninvolved and involved limb were tested. So the uninvolved limb is the black work loop, and the involved limb is the red work loop. And a few things that quickly pop out here. We see a loss of peak strength, so where the peaks of these work loops are occurring. We see a change in the joint angle where peaks are occurring, and we see really this loss of range of motion. So if nothing else, take home that these individuals have this significant functional loss, which is really driving some of the gaps in our understanding of how to treat them better. So we look at this injury pretty specifically in the context of military trauma, but it happens in a number of other circumstances as well. So this can occur secondary to open fractures, car, industrial or farm accidents, other related traumas, shark attacks, in that trauma category. Physiologically, it can also happen due to muscle resection with secondary to soft tissue cancers, or potentially loss in cleft palate and cleft lip conditions. So there's a number of instances where this is happening and where some of these treatments can be really beneficial. And of course, this is tremendously a burden. Here in the US, trauma itself is both expensive but timely expensive also. And in that military context, as I said, it's incredibly costly to the Department of Defense as well. So as I talk through this talk, I've kind of tried to set up how my lab and my collaborators' lab in the field really think about this problem. And it kind of falls into a couple of different categories. So first, different types of surgical or regenerative approaches that are really targeting that muscle that is lost. So how do we regenerate the muscle that is lost? There's another host of studies that are trying to approach the muscle that is left. So after that injury and that muscle is lost, how do we help the muscle that is still there, that kind of scaffold that is left? Also, understanding the pathophysiology of both the injury and that muscle remaining, and then how to support functional recovery, kind of in the context of the other three aspects. And I'll try to highlight which of these I'm talking about as I talk through some of our data. So how we evaluate this. So as the title of the talk alluded to, we use both small and large animal models of VML injury. And there are lots of models out in the literature as well. Latissimus dorsi, quadriceps, a whole host of muscle units that are being used. Somewhat collectively, it kind of ranges from this 10 to 50% of the muscle volume that is surgically removed in these models. But importantly, just to kind of keep in the back of your mind, that 10% injury isn't necessarily just causing a 10% strength defect. And we have good data to have shown that the strength defects basically are going to exceed that lost volume that we see there. So this is not a one-to-one relationship, but know that all of our models are do have chronic functional deficits that we will see. So in this, we have primarily broken this up in our lab, at least, into an anterior compartment model or a posterior compartment model. So our small animals, we're looking at the TA muscle, where we take a significant portion of that muscle out. As we move into the large animal model, we're taking this from the peroneus tertius muscle, which is analogous to the TA surgically, you'll see here. We've also started to use a posterior compartment model in our small animal model and are working to scale that up into the large animal. Importantly, as we think through this, in all of these instances, we have really focused on the function of the muscle, and through various platforms, it's fairly well conserved that we can evaluate nerve-evoked maximal function in these cases. So whether this is a small animal model or a large animal testing system, which I'll talk a little bit more about, our ability to look at force across the spectrum is consistent. And of course, I showed you just a little bit of biodex data. We also have that ability to move it into a more clinically relevant ability to answer that functional question also. Okay, so I have given you a brief introduction into what volumetric muscle loss injury is, keeping in mind that we have these irrecoverable functional deficits, this chronic loss of muscle, and what our clinical outcomes are. So as we approach these questions, mine and my collaborators' lab kind of think of it in sort of these physiology buckets. Is it neural dysfunction? Is it inflammation? Is it changes in force transmission? And I think we and others have kind of tried to tackle this in a number of ways. That said, none of these are independent. So even though I'm going to kind of talk you through some of our examples that potentially look at fibrosis, know that this very tangled physiology web is occurring, and none of these are happening in isolation. Also, before I get into some of our data, I'm going to significantly focus on nerve-evoked muscle function. That said, we use lots of tools to evaluate the muscle, and so do others in the field. But we try to keep high on our sort of standard of functional analysis, with our muscle strength being our key outcome that we're looking for, especially in our large animal models. Okay, so I'm going to take a quick detour before I get into the data and just talk to you a little bit about how we actually use Aurora's 890A platform to evaluate muscle function in these large animal models. And as Sydney alluded to, there are some handouts on the webinar platform. One of them will link to this Job video and paper where we have a very detailed protocol as to how we do this, as well as that video. And then the equipment manual straight from Aurora. So I'll kind of talk you through that picture that you see up there on the top. This pig is anesthetized. It is, you see the knee there, clamped in between two metal bars that's holding it steady at 90 degrees. The ankle and the foot are then attached to a foot pedal that once we stimulate the peroneal nerve, in this case, the hoof will push against that platform and that gets transmitted into a load sensor, and then we can measure the torque of this animal. So a couple of things. For this, you hopefully can see those couple electrodes that are there right by the knee, sort of near that fibular head spot. The peroneal nerve is fairly superficial there. That's the nerve we're isolating and stimulating in this case. And with this, we can glean a lot of information. We can get twitch torque, we can get tetanic torque, we can analyze lots of aspects of these force tracings as well. Again, remember that this is fairly well conserved. So I know that there's a lot of folks out there that are using this similar platform for small animal models. It's well conserved into the large animal model in what we're able to evaluate. How we have used it most. So we often take two approaches in looking at torque of these animals. So the first one on the one side of your screen here, we are very often looking at torque to angle relationships. So we will manually move the ankle joint into various degrees of plantar flexion from about 0, which is what you see in that picture there, to 50 degrees. We pretty consistently get our peak torques at about 30 degrees of plantar flexion, and that's fairly anatomical for the pigs in this sense. I will note that one thing that is very different with this 890A, at least the version that we have, it does not have a length controller, which potentially most people would be interested or used to rather with the small animal model. So we completely control where the angle of that foot pedal is as we are stimulating the nerve to evaluate function. We also pretty consistently use torque frequency, so delivering different frequencies of stimulation and looking at that relationship over our increasing frequencies. So in the bottom there, you see tracings of our increasing frequencies and then what that relationship ends up looking like. So we pretty commonly use both of these as we're evaluating function in these large animal models. We've also recently added on to this a little bit and validated, again, in that force frequency model, evaluation model, that these correlate well with our muscle activity. So using surface EMG electrodes, an example here of the 20 hertz stimulation and the 100 hertz stimulation from that same torque frequency curve, we get pretty strong relationship here between the two. So very analogous as we're thinking about other models or other methods to look at this function in these animals. Okay, back to our sort of bread and butter here as we're thinking about this story and kind of our web of physiology of what I'm going to talk through here. So I'm going to give you 4 or 3 parts of the story and then summarize. I'm going to give you a really simple example of how we have scaled up from the small to large animal model. I'm going to provide you a little bit more complex approach as we have thought about pharmacologically treating fibrosis. And then I'm going to explain some of our physiologic understanding that's supporting our current scale up, be that neural or metabolic, and then kind of think about where the field can continue to take this. So first, that simple example of scale up. This one actually hits a couple of spots on our sort of messy physiology web here. And it's really a combined treatment approach that we thought would highlight a number of the physiologic impairments following volumetric muscle loss injury. So we sought to evaluate if a growth factor, in this case, hepatocyte growth factor, HGF, loaded 3D aligned scaffold could support functional recovery. We started out in a rat model, and since this was meant here to be my most simple example, I'm only going to show you force and muscle regeneration. And down there in the middle, you actually see the 3D scaffold. These are sized to fit the injury that we are making. If we look at torque, in this case, in the rat model of our anterior compartment, we see pretty good or a modest improvement, both in that scaffold, which is in the far, in the orange, as well as a comparator, a minced muscle graft. But if we look at the bottom here in our deficit to control, we still see a pretty significant functional deficit. But I think importantly, the scaffold and the graft both had improvements, enough to allow us to potentially move this forward. Excuse me. One benefit of the small animal model is we can also look at muscle regeneration, in this case, looking at fiber number. And although we didn't see an improvement in fiber number with the scaffold, keeping in mind that function was our key outcome, we do decide to scale both of these up into our large animal model. And in somewhat of a pilot work, same setup, we're just evaluating the efficacy of this 3D aligned scaffold and comparing it to another treatment approach. And we get essentially similar results, modest improvements, somewhat chronically in function, although we still have this pretty significant functional deficit between them in comparison to those injuries that were left unrepaired. And I'll come back to this remaining gap here as we talk through a little bit more of this data. So that's the simplest approach that we would take to scale something up. From rat, make sure it is at least improving function in some way, scale that up to the pig and see what that looks like. So that's all well and good to do things, very straightforward like that. But I think more realistically, we're taking more complex approaches to scale things up. And I'm going to talk you through how we have done that now and thinking about mitigating fibrosis. So we know after these injuries, there's this huge accumulation of extracellular matrix that's occurring during the repair process. We know that this impairs healing, impairs regeneration. So we sought to somewhat tackle that side of this problem as we worked through this. And in thinking about this, before I get a little bit into the data, I will say that we've been really interested in using already approved approaches, already FDA approved approaches that are potentially repurposed for another use. So you'll see that here in a second. So this is a compilation of a number of studies where in our small and large animal models, we continually saw this incredible fibrosis that was developing after these injuries. In this case, anything that's this dark purple is very fibrotic, and the pinkish ones are the muscle that's there. As we moved into the clinical population with patient samples, we saw a really conserved view of this also. And in a lot of cases, essentially almost this tide mark of where the fibrosis and the healthy muscle or remaining muscle per se were occurring. So we tried to take a step back and understand what was happening here before we tried to potentially treat this fibrotic problem that we were seeing. So in thinking about this, to kind of go back in time just a little bit, we kind of went back to our rodent model to see how our data were potentially supporting what we were seeing histologically. And looking at a mouse model, so this is definitely a benefit of having that length control on the muscle testing platforms, in that we're not stimulating anything here, but under computer control, we can move that foot plate and see how much torque resistance there is as we move the ankle through a joint, a range of motion. And we consider this to be analogous to passive torque of the muscle within this animal model. So what you see in the graph there, that dotted line is actually the control. So usually that resistance to movement is quite low. But in our injured model, over time, we continue to see this increase in passive torque. So this was kind of one of our first indications that we're seeing this really impactful aspect of this fibrosis. And then just looking at the total collagen content of those muscles, it matched really nicely with this. We saw this increase in just general collagen content of the muscle over time, in this case up to 4 months after injury. And to us, this really indicated, supporting evidence of this increased fibrosis and increased stiffness. To move this forward a little bit, more recently, we went back and looked a little bit more, or started to look at least, at the composition of what this collagen looked like. So again, you're seeing just pan collagen, again, increasing over early time points after injury, so temporal increases. But what a graduate student in the lab did was actually take those time points and break it into different types of collagen to look at the response kind of broadly across them. So collagen 1, 3, 4, and 6, and looking at what those temporal relationships were over time. The graphs on the bottom are intentionally a little bit small. Oops, sorry about that. And if we, look at this top graph here in trying to look at these together, a couple of interesting findings kind of pop out as we have, spent a lot of time thinking about this. First, you see this huge increase in collagen 3 initially that eventually starts to wane, but this is an incredible increase in collagen 3. Our other more abundant skeletal muscle collagens here have this initial increase, they kind of plateau a little bit, but collagen 1, as that collagen 3 is decreasing, is also increasing fairly significantly. So to us, a couple of important aspects come out of this. So we do think that as time goes on, we are getting this accumulation of kind of more denser collagen, especially in the muscle that is left after injury. But I think it also gives us potentially something to think about as to what these sort of early changes are happening, sort of between that 2-week to 3-week time point. If this is providing us with good rationale to potentially use some of these time points and knowledge to intervene at fibrosis at different time points. So as we start thinking about this and continuing to move this forward, we went back to our pig model and essentially started to do the same thing. And we see a really conserved response between the just total collagen and that mouse model that I showed you and the pig model, again, suggesting that we have this increased stiffness. So on to what we tried to do and how we tried to mitigate this. So as I noted, we are very interested in already FDA-approved drugs that can be repurposed quickly. And in this case, we use the antifibrotic treatment nintedanib, which is a treatment for idiopathic pulmonary fibrosis. It targets a number of growth factors, fibroblast growth factor, platelet-derived, VEGF. So it's signaling through that mechanism. In early work in a mouse model, we know that it's not further impacting force, at least. So we're doing nothing or giving this antifibrotic, although it can decrease the fibrosis. We do have some force measurements there in our small animal model. And we knew that as we started off here in our pig model, if we delivered this treatment for 4 weeks, we could mitigate that increase in collagen. So this was enough support for us to start moving forward with this as a treatment model. So with just 4 weeks of treatment, we started the treatment immediately after injury to see if we could mitigate the development of this fibrosis. And because we don't have that length controller on the large animal model like we do the small animal ones, we use a different approach to look at stiffness in this case. So using shear wave elastography on an ultrasound-based system. I'll quickly talk you through this. If we look at the scale, the redder colors are going to be more stiff and the bluer colors are going to be less stiff. And we can easily see our muscle in this towards the top of the images, that's superficial to the limb, and then deeper down towards the bone as the image goes in. And we could quantify stiffness both in the muscle that was remaining, but also in that defect area, so where all that muscle was lost. And without getting too much more into that shear wave elastography, we found 2 important things. One, our antifibrotic was able to decrease our stiffness. So that's our dotted line in these graphs to the side. But also, when we did not treat it, we actually saw both stiffness increase in that area of injury, so in that muscle that's lost where we think fibrosis is just developing, but it also kind of expanded out into the rest of the muscle. So the muscle remaining is also having this secondary effect of these injuries. So onto our force. So in a non-invasive approach, we measured muscle function in the anterior compartment of the pig chronologically over this. So we tested before injury and then weekly after, up until that terminal time point. If we just look at the force deficit on the top there, we actually were, somewhat surprised by this. The antifibrotic actually reduced force more than just the injury alone. But if we corrected for the size of the muscle compartment in this case, we actually had fairly similar strength deficits if we normalize for muscle size in this case. So promising, but potentially still some questions remaining there as we look at the force for these animals. We also at the gene response within that muscle remaining, so at the fibrotic and wound healing gene response. In all of these, the gray triangles are the antifibrotic treated animals. And the take home from this is really that we preserved a lot of directionality of the healing responses. And we never, completely downregulated any of these pathways. And we think that's important, as there are important aspects of healing that need to happen, but also a lot of shared pathways between regeneration and wound healing in that case. Okay. So to sum up this short part here, we know that fibrosis is occurring, and this is actually very limiting to functional improvements as well as regeneration of the muscle. But where we're left with, this treatment is potentially beneficial. It's not doing any more harm, but I think we're still at a point where we're trying to optimize the delivery or timing. We did this treatment systemically, so working on some specifics there. We think it's important to keep it TGF-beta independent. I didn't go too far into the pathways, but TGF-beta, of course, plays an important role in muscle regeneration. And then I think the big question and looming question from this part and others is, how do we integrate this with other approaches? We were able to mitigate the fibrosis, but we did nothing to actually regenerate muscle. So something we're starting to think about sequencing or doing different treatments in parallel, and how we can improve function that way. Okay, so on to our next part, how we're starting to scale up using two pathophysiologic examples. So we're going to start with chronic neural dysfunction. And as we get into this, I just want to remind folks that full innervation of a muscle fiber is necessary, whether that is a regenerated muscle fiber, a repaired, one that's just remaining. We need innervation for function to occur. So we tackled this question in a number of different ways. Just to kind of give a very brief background on some of the aspects we're looking at here. We first started by just counting descending axons for the nerve innervating the muscle of interest, the muscle that's injured. We really see no difference in the number of axons that are innervating the muscle. This is occurring when we still have significant functional deficits. We feel that this sort of plateau that we're seeing here between 14 and 28 days is going to continue in time. So we started to ask a few questions on this. And the first thing we started with after that axon aspect was to look at the motor neuron. And not to get too far into the weeds on how we did this, hopefully you can appreciate that with these images down here on the bottom, we are able to specifically isolate and label the motor neurons of our muscle of interest. And then we quantified this looking at the number of total motor neurons and the number of motor neurons that were still communicating with our muscle of interest. And if we look at these black bars, those are the ones after injury. We are seeing that we have a significant number of neurons that are axotomized. So they're there, but they're not necessarily communicating well. But importantly, we actually don't see any motor neuron death. So between the ipsilateral and contralateral sides of the spinal cord here, our motor neuron number is remaining, although not necessarily functional. So I think to me, that's always been sort of cautiously optimistic that we have this retained but potentially limited capacity for reinnervation in this model. We moved that forward into the pig model as well. So you're seeing examples of pig spinal cord here at the level of interest for our muscle of interest, and not quite as eloquently as we can do in the small animal models. But we have continually maintained number of motor neurons here. So again, optimistically, we think that there's this strong retention or strong ability for retained capacity to reinnervate the muscle. So we decided to ask the same question at the neuromuscular junction. And in any given muscle, each of these muscle fibers, thousands and thousands of them, are going to have individual neuromuscular junctions that are controlling the electrical and chemical signals as these fibers are being activated. And pretty often we see there's just kind of a zoomed in example that you see on the side where we see nice pre and post synaptic overlap, the nerve innervating them. So the first thing we asked was, as we remove this muscle, what does that look like? And not surprisingly, we see lots of axons moving through here, but no neuromuscular junctions. There's no muscle fibers that are in that remaining area. So it's not surprising that we don't see neuromuscular junctions. So moving forward, the questions that we asked are in this muscle that's around where that initial injury are. What do those neuromuscular junctions look like? And this paper is published, so I don't want to get too far into the weeds on what we found with the help of a postdoc in the lab. But I'll kind of summarize a significant amount of work in one slide from this paper. So as we look over time, the most important thing that I think came out of this was we actually saw in that muscle remaining, the amount of denervated fibers continued to increase. So as time went on, we're seeing more secondary injury into the muscle where these fibers are denervating. And this was also paralleled with seeing increased sprouting and increased polyinnervation, which may have a degenerative or potentially a regenerative effect, but nonetheless, we're seeing kind of morphologically questionable aspects that are happening after injury. And while those NMJs that you're seeing on the side there are not necessarily representative, I will say that there is a large number of NMJs that go into these analyses, and often you'll sort of see very unique pathologic looking ones that you're seeing there. So the take home, besides this secondary denervation that was happening from this initial look at the neuromuscular junction, was that while we are seeing denervation, we sort of optimistically still think that there is this possibility that the neuromuscular junctions are there and able to support reinnervation. So more recently, we've kind of drilled down on this and tried to ask a little bit about specialized cells at the neuromuscular junction, in this case, terminal Schwann cells. So instead of just our pre and post synaptic staining, which you'll see in the red and green there, we've added on nuclei in the blue and then S100 to be able to co-localize those and look at Schwann cells. So usually there is a change in the number of Schwann cells after injury, and we thought that this could potentially be happening in parallel with our injuries and changes at the neuromuscular junction. So the first thing we started with, again, here's our example of that neuromuscular junction. All the arrows are pointing at Schwann cells. We looked at just the number of Schwann cells per neuromuscular junction after injury. And we really saw this delayed increase in Schwann cell number. So it remained pretty consistent. And then something happened at that 48-day or between 21 to 48 days that we see this huge jump in Schwann cell number. So that was a little bit curious that this was happening kind of in parallel with that change in or increase in denervation. So we took a step back and asked, what is supporting this? And one of the avenues that we looked down was really trophic factor activity. And one that I'm showing here, we have kind of a host of them that we've been looking at, neuregulin or NRG1. We saw this very initial and expected increase in neuregulin 1, only for it to drop down and just kind of spike back up a little bit there as this increase in Schwann cell number was occurring. And why that becomes important was, as I noted, we are looking for potential targets to support reinnervation. And one thing that we had sought to evaluate was repurposing a monoclonal antibody treatment that targeted neuregulin signaling through the ERB receptor. So the ERB receptors are very abundant within the neuromuscular junction. And these 3D renderings that you see at the bottom, hopefully on that purple one where you're just seeing the postsynaptic side, the ERB receptors are really abundant in the gutters of these neuromuscular junctions. So we've got lots of ERB receptors there. And although paradoxically, it's been shown that inhibiting ERB2 supports axon outgrowth. So we took a chance with the support of some of our neuromuscular junction data prior to this and decided to treat with this pharmaceutical to see if we could support innervation. Much like our other treatments, we started right away, and that continued through the time frame of after injury. So what did we find? I'll kind of start over here on the left and work my way across the slide. Unfortunately, we didn't see much of a difference in force. So inhibiting ERB2 didn't improve our force per se, but it did improve the neuromuscular junction. So moving up to the top, we can mitigate that significant denervation that we see there, and we can see that both qualitatively and quantitatively in the two graphs there on the top. We can improve those kind of pathologic aspects that we're seeing. So we can decrease fragmentation of the neuromuscular junction. So all in all, we think that this was actually really positive, even though there was no functional improvement. So again, somewhat in parallel to that fibrotic story is we can improve the physiology that's there, but we've somewhat lost our improvements in function. So as we think as I wrap this little section up, as we think about moving this forward, questions much like the fibrosis one, how do we optimize delivery or timing? Again, this was given systemically. And how do we optimize or think about treatment approaches working together? It is possible that we could improve innervation in this case with something like this or fibrosis, but how do we sort of jumpstart muscle regeneration also, or hypertrophy of the remaining fibers to allow these to work a little bit better, especially as we're thinking about scaling it up. Okay. And then I'm going to talk briefly about my last sort of pathology of this, our metabolic maladaptivity. So in line with some of the other stuff, we were kind of left with a few questions of what the metabolism of this muscle remaining looked like, and how it's impacting the whole animal or potentially the whole person in some of these cases. So kind of our initial steps into this, we first just looked at the mitochondrial network after injury. So the mitochondria in these cases are green. Using some math on this, which I won't get into, we can actually look at the alignment of the mitochondria, which are in our control panel there on the far side, usually very well aligned. After injury, we realized that this became very dysregulated. And the farther we moved out from the injury, it was still very dysregulated. And even, as far proximally as we could on the muscle could be, we were still seeing significant dysregulation compared to the control injury. So this was kind of our first question as we were thinking about the mitochondria. So we started off, very simply saying, just kind of what's the mitochondrial content? What does that look like? And we, there wasn't super convincing data here, slight increase rather in mitochondrial content. But we realized that there was, in parallel with this, really this reduced oxidative capacity of the muscle. And if, more recently, as we've looked at this, looking at mitochondrial function more specifically, we see that although there's potentially an increase, we see this decrease in the respiratory states of the mitochondria over time. So although we have more mitochondria potentially, they aren't functioning quite as well. We know they're very dysregulated. So it kind of pushes us down this pathway of how can we potentially treat this. So kind of thinking back to that functional question, starting to look at conductance of the mitochondrial membrane, and started to provide us some insight that maybe we have this impaired fuel oxidation that's occurring locally. So to us, this kind of drove a potential question as we were thinking about it, and we moved back a little bit into the large animal model, or into the whole animal model, and thinking about the metabolism. So the first thing we did was just make sure that they just weren't less active over time. So starting before injury and then multiple weeks after injury, they're still just as active as they were before. So it's not a disuse model per se. And then we looked at respiratory exchange ratio. So, and to kind of give a brief look here, we would be using more lipids as fuel. The lower we are to 0.7, more carbs as fuel, the closer we're getting to 1. And realistically, we generally have a mix in between these. As time goes on, hopefully, if nothing else, you can appreciate that our purple and green lines here are starting to just kind of get condensed. We don't have that ability to kind of fluctuate between fuel sources as that control animal did. And if we quantify this time course of respiratory exchange ratio, we can look at the average RER in the active phase and the inactive phase, and really look at the difference in them, so that delta RER. And we think that this implicates that there's this kind of loss or decrease in carbohydrate oxidation that's increasing lipid oxidation. So this small injury, which is not impacting activity per se, is having pretty significant whole body metabolism outcomes. So where does this put this? We are at the point of just starting to scale this up into the large animal model. And questions that we have now are, can we even optimize a treatment? Can we somehow correct the mitochondria to make the muscle a little bit more responsive to oxidative changes? And we think that this has been a really important avenue for us to go, just so we can think about long-term health in this clinical population. And again, we are strategizing ways to really integrate this with other approaches, be that antifibrotics or neural enhancing drugs. Okay, so I talked you through a simple example, a little bit more of a complex one, and 2 pathophysiologic questions that we have. So in the couple minutes that I have left, I'm going to talk kind of about the next steps. So if nothing else, I hopefully have convinced you that this is a really complex problem, and it impacts the muscle that's left, the muscle that's gone, but also that we've really stressed how to improve function. And back to how we improve function. As I started to get this talk together again, I went back and pulled together all of the things that we have looked at in this large animal model. And I've shown you some of the data that you see here. One thing that's been really nice using this muscle testing system from Aurora is we can actually compare across our studies pretty well. Our reproducibility is very high. And like I said, you've seen some of these. If we look at the injury that is just left to repair on its own, that's our dark blue line that we're seeing down here. Some things are working. We are seeing some functional improvements, but I think we still have this pretty wide unmet therapeutic range where potentially overlapping or in parallel treatments may be able to kind of close some of that gap. I think that also leaves us with a really possibility for novel and exciting treatments to potentially come in and also fill that gap in there. So as we keep working, us collaborators, the field, we really think that the complexity of this problem is going to require a multidisciplinary approach, and how we can correct the physiology and promote a regenerative permissive environment is going to be really important in that case. And with that, I just want to thank first my 2 longtime collaborators, Jared Call and Ben Corona. They both contributed to a lot of the work you saw here, as well as members of their individual teams. My current lab and my past lab members that you see on there, Matt and Chris at Aurora, Sydney at Inside Scientific for her help, and of course the American Physiologic Society and our host of funding that has helped with all of this. And with that, I am going to hand it back to Sydney here for a minute.
Yeah, great presentation, Sarah. Thank you so much. Thanks again for that awesome presentation. As new questions start rolling in, let's start with our first couple that we received. Dana has asked, in the metabolic situation, did you correlate mitochondrial dysfunction with decreased contractility measured in the muscle?
Yeah, so in the small animal, we do have some of that data, and actually, I don't think we've correlated specifically, but our small animal data was just recently published, and it is there. I think one of the things that we have worked on with the metabolic side of things is trying to understand, and I didn't present this data here, why it's actually not the muscle remaining is not responding to known stimuli that should improve mitochondria. So things like exercise or overload stimulus, we aren't seeing that response here. So we're trying to understand a little bit how those relate to the function, but correlate specifically, I don't think we have. Now that I say that.
Okay, perfect. Thanks. Our next question from Yuri, does movement affect the development of collagen in the muscle after an injury?
Yeah. So we have done some work where we tried an approach where we just used passive range of motion, to see if we could change the trajectory of that. And we haven't seen much impact on that, but we haven't explicitly looked at different collagen isoforms per se afterwards. We, anecdotally, we don't have this data explicitly, but we don't see a lot of movement impairments, at least in our animal models after injury. So we don't think that's contributing to what we're seeing there.
Okay, great response. Thanks. All right, the next one on my list, Kevin, he commented, very nice presentation. How important is the location of the injury to the outcome? So in particular, in terms of damage to the vascular system as well as nerve branches.
Yeah, I think that's a great question. I actually don't know that that's been looked at specifically. We think about it a lot in our lab. We, try to standardize these models as best as possible for reproducibility. But of course, clinically, this is a really heterogeneous injury type, right? You won't see two that are presenting in the same way. But I think easily, if this injury were potentially right where the nerve stump is potentially moving, inserting into the muscle, that could be more functionally impactful than something that is, in the middle third or the bottom third, or in the same way that something that maybe impacts the myotendinous junction could potentially be impacting there. So I I think there is a regionality of it. We have really stressed using functional deficits as our outcome to be able to kind of get around that a little bit, but I don't think there's good data to support it at the moment.
Okay, thank you. Albert has asked whether you've applied this type of functional analysis in porcine models of hind limb ischemia or denervation.
Yeah, so we have not done it, but I do know there's a group who has used the large animal functional testing in the ischemic model. So it definitely is out there. I think for us, for the large animal side of things, we focused really on this injury, but there's a whole host of things that you could be using these for. Be that normal physiology, a lot of the neuromuscular disorders that are, moving into pig models. All of these would be great options to use that large animal testing system. But we have not specifically. We've really focused on BML.
Okay, that makes sense. Okay, let's jump to this one here. Noah has said, is there a persistent loss or reduction of muscle blood flow / capillary network to these injured areas? Or sorry, in these injured areas? Does this figure in the overall picture of failure to regenerate muscle function?
Yeah, so that recovery of vasculature is, of course, equally as important. That was one of the physiologic aspects that I just didn't touch on. We don't focus much on the vasculature. It's a little bit hit and miss, the data that is out there. I don't think it's been well evaluated. But yes, you absolutely need the vasculature to respond, be that just for blood flow, for paracrine factors, growth factors that are needing to get to the area. It will impact functional recovery, not having that as well. And keep in mind, these injuries are I focused really on the muscle fibers, that loss of fibers, but it's taking all the endogenous parts of muscle, right? The scaffolding, the extracellular matrix, the vasculature, that's all being removed in these injuries. So yeah, all of them are important to recover in that sense.
Okay, fantastic. So Stan has asked, can you comment on VML injury in aged, equivalent to, say, a 60-year-old in humans, in aged animals, and the ability to obtain aged large animals?
Yeah, that's a great question. So we have not done any work in aged animals. I know that there is a group who does look at them, who kind of focuses their research questions a little bit more on that side of things. And in part, they're trying to look at, is the regenerative response different in these animals? So yeah, we have always used adult small animals as well as, adult pigs as well. We don't have any data on that. I will say, depending on the aged animals, depending on what strain of pig you are using, there will be some limitations. As probably people realize, pigs grow a lot. Everyone's probably been to their state fair and seen these giant pigs, or maybe they grew up on a farm. They can get really big. So at the moment we are considering shifting into maybe the mini pig model. Not that they just don't grow quite as much, they still get plenty big. But I think that would be a limitation if you're thinking about using the large animal model in an aged pig. Is they're going to be quite large in thinking about that growth curve, and the functional range, at least, there's lots of aspects, right? There's the anesthesia, there's the housing, like so many aspects will fit into these really large ones, but there is an upper limit per se on the functional apparatus that in that 890A. I think the largest animal that we've used is about like 130 kilograms. So we've definitely used bigger ones, but they will just continue to get bigger. So I would hesitate to say in the large animal model to get aged ones.
Okay, great information. Thanks. Okay, let's see here. Michael said, along the lines of Uri's question, can you make a distinction between movement and loading?
That's a great question. Other people can. We do not have that capability in our lab at the moment. But yeah, you could have force plates, and we've talked a lot about this. There's some treadmills or walkways, right, that'll have parallel force plates, and you could isolate which limb was loading in which situation. We just don't have the data. Anything that I showed you is just activity. But yes, there's always the possibility that they are just not loading the limb that was injured. That definitely is there. We just don't have the data, but you could do it.
Mm-hmm. Okay. Awesome. Let's address this next one. Sorry, let me see here. Okay, I think I'll go with David's question. Is there a difference in the sex of the animal's recovery or response to VML injury? I know that this is usually a big question in a lot of studies.
Yeah, so with the data that we have, recently we've started matching our study populations, males and females, and we have not seen a difference. That's not to say that it's not there. Most of the data out there has been on males. And I think clinically, all of the case studies that are out there are all males as well. I would hesitate to say there's absolutely no difference, but we haven't seen anything yet.
Okay. All right. Great response. Thank you. I think I will make that actually the last question. So I just wanted to say, Sarah, thank you so much for your time and expertise today. It was such a pleasure to have you with us.
Yeah,
And I
thank you all
hope
so
you
much.
enjoyed yourself. In closing, we hope you enjoyed this Inside Scientific webinar produced in partnership with the American Physiological Society and sponsored by Aurora Scientific, and I hope you enjoy the rest of your day.